So again, looking at cells and talking about cellular anatomy, we've looked at prokaryotic cells, right? Remember, bacteria and archaea are prokaryotic cells. Prokaryotic cells are small, they're simple, remember, one-room studio apartment. Now remember, the best place to start when you look at a cell is looking at the plasma membrane and working your way in and out from there when you're concerning yourself with anatomy. So plasma membrane defines our cell. That's chemically inside of it is the cytoplasm, and outside of that is the exterior of the cell. Remember, in prokaryotic cells, they have a cell wall, and we want to add on to our information about prokaryotic cells, and that cell wall is made out of a substance called peptidoglycan. And so that is one substance that you can always test for to be like, is there bacteria in the solution? And peptidoglycan is going to be associated with bacteria because that is what their cell walls are made out of. We'll come back to the function and why cells have cell walls in just a little bit. But for now, let's move on from the simple cell that is a prokaryote to eukaryotes. And when we look at pictures of eukaryotes, immediately we can see that this is a much more complicated system. So we're going to break down our eukaryotic cells into two major classes, animal cells and plant cells. And the vast majority of organelles that we're going to see are in both of these types of cells. So assume that something is in both kinds of cells, is in all eukaryotic cells, unless I mention it otherwise. So we're going to start with the center of the cell and the organelle that is the most obvious using a light microscope, and that is the nucleus. So this is going to be this big purple thing right in the middle of that animal cell. All right. So when we look at that nucleus, you can see it's cut away, and you can see inside it, and inside is what's called the chromatin. And the chromatin then is all of the DNA that is inside the nucleus. You can see it's got an envelope, so it's actually got two membranes that make up the nucleus, and then there are pores in that nucleus that allow things to move in and out of it. This is a room inside our eukaryotic cell. And this room, the purpose of this room is to store the DNA and keep it safe. All right? So the chromosomes, you've heard this term before possibly, the chromosomes are large pieces of DNA, whereas prokaryotes just have a naked chromosome out there in the cytoplasm. The chromosomes of eukaryotic cells are always contained inside the nucleus. Now, another component that is found in all eukaryotic cells are the ribosomes. And in this picture, they're so tiny that you really can't see them very well at all. So you see the word ribosome over on the right, and it's pointing to what? It's pointing to those little red dots. So if we zoom in on those little red dots, we can see that there is a specific shape to that ribosome. Now, we talked about ribosomes before in prokaryotic cells. So ribosomes are actually a feature that we find in all cells, but eukaryotic ribosomes are a little different than prokaryotic ribosomes. And we'll talk about that again a little bit later as well. But a ribosome is made of two subunits, a large subunit and a small subunit. And for now, what we really want to remember is that this is the site where proteins are built. Right? So this is kind of the little protein factory that is necessary to take the information that ultimately is going to come from the DNA to build a protein. Now, they're interesting in the fact that the ribosomes themselves are made of proteins and RNA, where the RNA is actually part of the structure itself. Right? It's actually catalyzing the reactions to a great extent. So the ribosomes can be found in two locations in a eukaryotic cell. They're either going to be free-floating in the cytoplasm, or they can be attached to another organelle called the ER, or Endoplasmic Reticulum. And so as we talk about the ER, what we're going to do is begin a journey of a protein through what's called the endomembrane system. And so the red arrows in this picture of a cell show you this folded network of membranes that are studded with ribosomes we call rough ER. This is where many proteins are formed, where the ribosome attaches to the rough ER and builds a protein and injects it to the inside of this new room in the eukaryotic cell. So this is yet another room in our eukaryotic cell, the endoplasmic reticulum. And we're going to follow the journey of that protein through the different organelles that we collectively call the endomembrane system. So let's start with the ER, endoplasmic reticulum. Endo, inside, plasm, like a cytoplasm. So we're inside the cytoplasm, reticulum. Reticula is the Latin derivative for net-like. So it's a network or a net-like structure of membranes inside the cytoplasm. That's the ER, endoplasmic reticulum. And what we can see here is a ribosome that is attached to the ER, making it rough. So literally it's studded with all those little ribosomes, and if you were to run your hand on it, in a metaphorical sense, it would feel rough. And so those ribosomes, being the sites of protein synthesis, are actually making the polypeptides, making the proteins, and injecting them across the membrane into the interior of the ER. So we talked about the eukaryotic cell partitioning the cytoplasm into different quote-unquote rooms. Why do we partition the ER? Because in the ER are going to be enzymes that are going to help to glycosylate that protein. So proteins that go through the endomembrane system are going to be decorated with sugars. We're going to start to combine our biomacromolecules so those proteins function correctly. We're going to start to add sugars to them and make a glycoprotein. Now, the thing about membranes is that they're fluid. And again, we'll come back to the idea of the fluid mosaic model for membranes. But literally, you can pinch off a piece of that membrane like a soap bubble, right? And so the main body will remain intact, and you can kind of twist off or pinch off, bleb off a little piece of it, and it becomes what's called a secretory vesicle. So a vesicle is now like a little tiny bubble from the ER, and it's containing the inside of the ER inside of it. So you have the proteins that were inside the rough ER can now be inside a secretory vesicle that can move to other parts of the cell. So again, we see the rough ER with the dotted ribosomes attached to it, and we can see that it's connected to the nuclear envelope. But the rough ER is going to be our starting point of our endomembrane system. Now we can see, here we go, rough ER, transport vesicle from ER. The arrow starts to move towards another network of membranes called the Golgi apparatus, or the Golgi bodies. And we want to think of these as kind of like a stack of pancakes. And so you can see in the electron micrograph on the right, the membranes do look like a stack of pancakes. And then the cartoon rendition on the left, you can see that kind of looks like that. And that's what the Golgi is going to be characterized by. You're going to see those thin stacks right next to each other. That's the Golgi for sure. Now, what happens in the Golgi? The Golgi is going to be kind of our processing plant, right? So we're going to talk about the proteins that come into the Golgi, and they're going to be further polished with sugars. Right? So we're going to modify those sugars a little bit, but also we're going to sort the proteins. And the proteins can go off into different locations from the Golgi apparatus. So one such location for the Golgi is going to be the plasma membrane. Another location is going to be vacuoles, and a third location is lysozymes. Right? So let's look at this kind of breakdown in a flow diagram. So again, the path of the protein through the endomembrane system is going to start by synthesis on a ribosome on the rough ER. Through a transport vesicle, it's going to travel to the Golgi body. In the Golgi body, it's going to be further polished, further glycosylated, and then sorted, sent to either a secretory vesicle, okay, which is like a transport vesicle for secretion, merge with the plasma membrane, and then be secreted to the exterior of the cell. Right? So this is how hormones get dumped into your blood to travel throughout your body, like adrenaline. So when you go into a flight or fight response, those cells that make adrenaline are dumping that protein into your blood from secretory vesicles, fusing with the plasma membrane, and then dumping it into your circulatory system. The Golgi body can also send vesicles that become lysozymes, or lysosomes, sorry. These now are going to be packed with proteins that are going to be involved in breaking down food that the cell eats. And we'll take a look at that in just a second. And then finally, a transport vesicle can take proteins from the Golgi body to vacuoles, which are kind of storage locations or other locations in the cell. Now, let's take a look at lysosomes for a starter. So lysosomes now are small membrane vesicles containing digestive enzymes. So those proteins that are produced in the ER that are going to ultimately chomp up food are going to be shuttled towards a lysosome. Now, how does that work? So we'll talk about how things move into the cell a bit later, but there's a process called phagocytosis, and this is cellular eating. And so the plasma membrane is going to engulf a bit of food particle and make a food vacuole. So that vesicle inside the cell now will be ultimately fused with lysosomes. So lysosomes coming off the Golgi apparatus full of those digestive proteins are going to fuse and break down that food particle. We are doing now hydrolysis. We are breaking down proteins and lipids and carbohydrates and nucleic acids into their component parts that then you can use, or that cell can use, to build its own nucleic acids and proteins and carbohydrates and what have you. All right, so that is kind of really the one way of cellular eating. Now, another thing that the lysosome will fuse with is a process called autophagy. So autophagy is when you recycle organelles. Now, we haven't talked about mitochondria yet, but that's like one of the main targets for autophagy. You're going to take one of the rooms in the house that's kind of broken down and well used and actually recycle it so the cell can reuse those parts. All right? So that process is all involved with lysosomes coming from the Golgi apparatus. Now, the other thing that we want to think about are vacuoles. And vacuole is a general term that we use for storage. Basically, when we don't know what to call something else in the cell, we call it a vacuole, except for one major organelle in plant cells, and that is the central vacuole. When we talk about the central vacuole, we're talking about a very specific, very large organelle in plant cells. And you can see it here as that large clear bubble in the center. So it's a central vacuole, and the tonoplast is the specific name for that membrane that makes up the central vacuole. Otherwise, vacuole is a very, very general term, and we use it to store stuff that we don't know how to break it down or can't break it down, or foodstuffs for later. But in that plant cell, again, that central vacuole is going to be very important for maintaining what's called cell turgor, and we'll talk about that later. So finally, we look at the endomembrane system. We can see proteins moving from the rough ER to the Golgi apparatus, either being secreted to the plasma membrane, into vacuoles, or into lysosomes. Now, there's one other thing on this slide that we need to mention, and that is the smooth ER. So the smooth ER is connected to the rough ER, but it is not dotted with ribosomes. And so the smooth ER is kind of like a different chamber because it serves a different function. And the smooth ER is smooth because it doesn't have those ribosomes, and it's involved in a lot of metabolic processes. So it sequesters enzymes for synthesizing lipids and for detoxification. So, for example, your liver cells have lots and lots of smooth ER because your liver is what kind of the organ in your body that cleans up your blood. So we need lots of enzymes, lots of proteins in there that are going to break down the bad stuff that's in you and process it. All right. So we will spend another recording then on the other remaining components of the eukaryotic cell that we are interested in knowing about.